Hi, I am Tarun Tyagi from Yale School of Medicine, and today I'm going to describe the main findings of our JEM paper entitled Platelet-derived TLT-1 promotes tumor progression by suppressing CD8 T cells. Anti-tumor immunity primarily depends on T cells, where normal functional healthy T cells are able to do tumor cell killing, while dysfunctional T cells, which are induced by immunosuppressive mechanisms because of the tumors, they are not able to kill. In fact, they help tumors to progress. Now, modern day immunotherapies are based on a rescue of these dysfunctional T cells into anti-tumor functional T cells. And these mechanisms are highly complex. As platelets can interact with a variety of immune cells, we ask if platelets have any role to play in these immunosuppressive mechanisms. We focus on TLT1, which is also known as TREM-like transcript 1. It is a platelet-expressed abundant protein, an extracellular region, a transmembrane region, and a cytoplasmic region. Upon activation of platelet, this comes on the surface and can be released. It has an IgV domain, which is interesting because this domain is shared by other immunoinhibitory proteins released by other cells. We hypothesize that platelet-derived TLT-1 influences anti-tumor immunity. We looked at the freshly isolated platelets from the non-small cell lung cancer patients and found significantly increased levels of platelet TLT-1 on the surface, in the mRNA level, and the soluble TLT-1 levels in blood. All were significantly higher. Now, the next big question was whether this is because of the tumor or because of the other secondary factor in this patient. So we used just non-small cell lung cancer PDX-derived mice. Patient-derived tumors were grown in the humanized mice, and the mouse platelet TLT-1 levels were found to be increased. Importantly, the tumor size increase was positively correlated with the platelet TLT-1 levels in these mice. Next, using a Syngeneic mouse tumor model study, we assessed the effect of recombinant TLT-1 injection when given to mice. After 1 week of tumor growth, mice were given either vehicle or the recombinant TLT-1, and the effect on T cells and the tumor growth was assessed. What we found was the T cells, the activated CD8 T cells, were reduced significantly in these mice in the blood and the tumors, and the tumors were grown excessively and significantly increased in these mice, which were given recombinant TLT-1. The direct evidence of the effect of platelet TLT-1 on the T cells from the patients came from this co-culture study in which the T cells and the platelets were isolated from the non-small cell lung cancer patients, and they were co-cultured. What we saw was platelets were able to induce the T cell suppression in this study, and the use of anti-TLT-1 antibody was able to rescue the platelet-induced suppression in these patients. Interestingly, the survival of these patients in our cohort was inversely correlated with the platelet TLT-1 levels, what we had measured. To summarize, reprogrammed platelets in NSCLC are able to suppress CD8 T cells by releasing soluble TLT-1, which we found was able to bind to the CD3 epsilon subunit on the T cells, which is causing the tumor progression. Clinically, TLT-1 can be a promising therapeutic target in non-small cell lung cancer and other cancer types. The ability of TLT-1 to regulate T cells can have wider implication in a variety of immune disorders. Thank you.